Hello, everyone. Thank you all for joining me today, and welcome to February 2022 eJournal webinar session. The title of today's article is Molecular Testing Results as a Quality Metric for Evaluating Cytopathologist Utilization of Atypia of Undetermined Significance Category for Thyroid Nodule Fine Needle Aspirations. Authors are Dr. VanderLaan and Dr. Nishino from Beth Israel Medical Center, Harvard Medical School, Boston. This article was published online on September 2021 in JASC Journal. I am Dr. Seema Khutti. I am Assistant Professor at Zucker School of Medicine, Hofstra, Northwell, New York. I practice gynecologic pathology and cytopathologies, and I have no disclosure. The objectives of this presentation is to review practical issues related to intermediate thyroid FNA cases, especially risk of malignancy and molecular correlation, to investigate the utility of combining AUS utilization rates with Afirma GSC results as a quality improvement metric for describing cytopathologist practices, to provide a novel framework for guiding cytopathologists' diagnostic threshold for AUS category based on their relative rates of GSC suspicious results. The Bethesda system of reporting thyroid cytology has provided a uniform reporting system for thyroid FNAs, with each diagnostic category carrying an implied risk of malignancy. The aspirates classified as AUS are heterogeneous, and AUS category continues to be an area of focus for standardization and quality improvement. The risk of malignancy ascribed to AUS nodule is approximately 10 to 30%. It's low, but it's not quite insignificant. Management decisions typically require integration of multiple factors such as patient characteristics and preferences, sonographic features, repeat FNA cytology, and recently ancillary molecular testing results. The Afirma classifier GSC combines high throughput RNA sequencing technology with machine learning algorithms to risk stratified cytologically indeterminate nodules as either having a benign or suspicious gene expression profile. Cytologically indeterminate nodules, which are AUS or follicular neoplasms, if they are classified as molecularly benign, are usually considered to have a low cancer risk approaching to that of a benign nodules. In contrast, however, indeterminate nodules with suspicious Afirma results are estimated to have a risk of cancer approximately 50%, and for which a diagnostic lobectomy would be considered. Periodic monitoring of AUS rates by cytopathology laboratories may be helpful for quality improvement. And the Bethesda system proposes maximum AUS rate of approximately 10% as a benchmark. However, for laboratories or individual cytopathologists that exceed these targets, it is not quite readily apparent how practice patterns should be modified in order to lower their AUS rate, as the AUS rate alone is inadequate to determine the root cause of AUS overuse. Theoretically, excessive AUS calls are derived from two scenarios: either overcalling the aspirates that are better classified as benign, which is category 2, or undercalling the aspirates that are better classified as follicular neoplasm, which is category 4, or suspicious for malignancy, which is category 5. In this study, authors investigated the utility of combining AUS utilization rates with Afirma GSC results as a quality improvement metric for describing cytopathologist practice, a novel framework for guiding cytopathologist diagnostic threshold for AUS category based on their relative rates of GSC results was also proposed. Moving on to materials and methods. thyroid FNAs performed and evaluated from December 2017 to July 2021 were included in this study. At Authors Institute, Afirma GSC testing is a two-stage process. First, patients with thyroid nodules meet criteria of FNA-B, undergo FNA for cytology only. And then after, only the cases with the results of either category 3 or category 4 undergo repeat FNA. And at that time, samples for cytology as well as Afirma GSC testing are collected. Until January 2021, Afirma testing was sent as long as the repeat FNA cytology was classified as category 1, 2, 3, or 4. However, starting January 2021, their institutional policy for Afirma testing was updated to exclude the testing of the nodules which were classified as benign, which is category 2 on repeat FNA. And the most AUS nodules with Afirma suspicious results were then referred for surgery, which was diagnostic lobectomy, whereas majority of the AUS nodules which came with Afirma benign results were followed up clinically depending on patients' preferences and clinopathologic features. Data was then extracted retrospectively from LIS, including the total number of thyroid FNA, diagnostic categorization, and signing out cytopathologist. The results of Afirma GSC testings were also retrieved. The rate of Afirma GSC suspicious test results were calculated using the subset of cases which were classified as AUS on a repeat FNA. Statistical analysis and linear regression was performed using Excel, and statistical significance was established as a p-value of less than 0.05. So these are moving on to the results. This is a flowchart tabulating thyroid FNAs and diagnostic category utilization for a cytology laboratory for the study. As we can see here, during the study period, the cytopathology laboratory interpreted approximately a total of 3,008 thyroid FNA cases, and the global AUS rate for the lab was 22.3%, as we can see here. The rate of suspicious Afirma genotypic classifier results was calculated only for the results which had AUS on repeat FNA, which comprises of total of 167 cases. Moving on to this next figure, this is a stacked bar chart illustrating the distribution of thyroid FNA interpretation based on Bethesda diagnostic category, stratified by individual cytopathologist and a lab average. So as we can see here, the lab average AUS global rate was 22.3%, and the AUS rate for 9 different cytopathologists ranged from 11.6 to 39.3%, with the number of thyroid FNA cases reviewed by each cytopathologist ranged from a total number of 82 cases to 788 cases by this individual cytopathologist. And of total 672 thyroid FNA diagnosed as AUS, Afirma GSC testing was performed on 167 cases which had a repeat AUS on second FNA. Moving on to this next figure, which is a stacked bar chart illustrating the GSC result, which was either suspicious, benign, or non-diagnostic, or either the test was not performed, stratified again by individual cytopathologist and a lab average. The Afirma GSC results were suspicious in 48 cases, which comprises of 29% of the total cases. And majority of the cases, as we see here, which is 117 cases out of this second subset, which was 167. 117 cases had a benign Afirma results, which comprises of 70% of the total number of cases. And only 2 cases had either non-diagnosis or non-diagnostic test results, or either the test was not performed, which is 2 cases, which comprises 1% of total case volume. Afirma tested subset of AUS nodules for each cytopathologist. Again, as we can see here, range from total number of 4 cases to 37 cases, which was quite a wide range. And the suspicious rate, or the Afirma suspicious rate for individual cytopathologists, again, ranged from 5% here to 67%, which was a wide range. The histologic outcome data was then compiled for AUS nodules with suspicious Afirma test cohort only, as majority of the AUS nodule with benign Afirma test results were followed up clinically. And of those 48 cases which had Afirma suspicious results, histological outcome data was available in 41 cases, which is approximately 85% of the cases, of which 12 cases had a benign outcomes, which was either follicular adenoma or hyperplastic nodule in 29% of the cases. NFP was observed in 10 cases, which comprises 24% of the cases, and malignant outcomes was present in other remainder of 19 cases, which were all papillary thyroid carcinoma, 13 of which were follicular variant, 5 were conventional type, and 1 was tall cell variant. For the resected AUS nodule with suspicious Afirma results, the calculated risk of malignancy was 46%, and the calculated risk of surgical disease, that is malignancy plus NFP, was 71%. So to determine whether a relationship exists between an individual cytopathologist overall AUS rate and the rate of Afirma suspicious calls in subsequent Afirma tested subset, data was, this data were complied as a scattered plot, as we can see here. But as we noticed, there was no correlation between these 2 variables as identified. So these blue dots are individual cytopathologist, and the orange dot is the cytology lab average. Moving on to the discussion. So as we know, AUS category is very subjective and is prone to be overused. And providing feedback to cytopathologists about their practice patterns relative to the lab average and published benchmarks can promote judicious use of AUS category. As we discussed earlier, the suggested maximum AUS utilization rate is approximately 10% as a target. And the raw AUS rate is a relatively easy metric to compile. However, the AUS rate alone is insufficient to explain the root cause of potential AUS overuse. And determining the outcomes of AUS nodules provides some insight. However, most AUS nodules are not resected, and the metrics derived only from the surgical cohort are limited by verification bias. And hence, author investigated if molecular testing results of AUS FNA could complement the AUS rate as a performance metric for individual cytopathologists. There was no strong lab-wide correlation between the Afirma suspicious or Afirma testing results and AUS rate. However, author recognized parallel between the thyroid FNA data and those that implied high-risk human papilloma virus positivity rates as an objective quality metric for cervical pap test. As we all know, through an elegantly designed thought experiment, Dr. Sebus and colleagues have combined pap test ASCUS to seal ratio with high-risk HPV positivity rates of ASCUS PEP test results to establish a framework for providing a useful feedback to an individual cytopathologist in comparing their practice patterns to target norms. And so authors have compared these two tests as an Afirma GSC test and high-risk HPV test have some similarities. Both provide binary outcome, both have high negative predictive values, and are used clinically to guide the management of patients with indeterminate cytology. So authors applied the similar conceptual framework to thyroid FNEs in the current study. Variations in practice pattern among individual cytopathologies likely explain the absence of lab-wide correlation between the Afirma GSC result and AUS rate. And hence, authors propose that individual cytopathologists' Afirma suspicious rates can provide an additional dimension to their respective AUS rates, as we can see, as we'll see in next few slides, and offer an insight into the ways to adjust the diagnostic thresholds. So this was a figure which demonstrates this conceptual framework for combining AUS usage rate and molecular testing results to explain cytopathologist practice patterns and how to bring their practice in line with desired targets. So here in this left top figure A, as we can see here, most practices have AUS rates that exceed this target rate of 10%. However, how do we bring our practice in line with this target is to be thought. So moving on to this figure B, this is an example here where the individuals with high AUS rate is high compared to our target, but the Afirma suspicious result, which is GSC S rates, are relatively very low. So these are the group of individuals who are likely overcalling their thyroid Bethesda 2 nodules. Moving on to this figure C here, these are the individuals who has both a high AUS rate and high Afirma suspicious rate. They are the individuals who are likely undercalling their follicular neoplasm, which is category 4, or suspicious for malignancy, which is category 5 FNA as AUS. So these are those individuals who are likely undercalling compared to these individuals who are likely overcalling. The next group of cytopathologists, as we see here in this figure D, are the individuals with a very high AUS rate, as we can see here, these three individuals, but at the target for a lab average of a GSC suspicious rate. These are the individuals who are likely overcalling their thyroid Bethesda 2 FNA as AUS, and as well as undercalling their category 4 and category 5 FNAs as AUS. So there is both a combination of overcall as well as undercall. While and the optimal way to counsel these cytopathologists in this circumstance is not clear-cut, it is possible that both the upper as well as the lower boundaries of their criteria for using AUS needs to be constrained. It's unclear if lab or individual cytopathologist AUS rate could ever be considered too low. AUS rates below a 10% generally would not warrant correction. However, in a hypothetical scenario where unusually low AUS rate could be associated with an unacceptable increase in false negative cytology results, that is, increase in risk of malignancy associated with benign nodules, or decrease in risk of malignancy associated with the nodules which are categorized as 4, 5, or even 6. The Afirma suspicious rate could potentially help explain the source of AUS underuse. For instance, a cytopathologist with a tendency to classify cytologically borderline aspirates aggressively as 4, 5, or 6, lowering their AUS, while at the same time reserving the AUS category for a smaller proportion of the cases better classified as thyroid Bethesda 2, would on balance have low AUS call rate and a low Afirma suspicious rate. In contrast, low AUS rate coupled with high Afirma suspicious rate could indicate a tendency to overuse the Bethesda 2 category for the aspirates better classified as AUS, while reserving the AUS category for smaller proportion of the cases that may be better classified as 4 and 5, increasing the suspicious Afirma rate. So as we know, or we might have noticed, there are a few limitations of this study. The Afirma GSE testing has an approximately 70% specificity for carcinoma and 50% positive predictive value among AUS nodules. And therefore, Afirma suspicious results are an imperfect endpoint for assessing the AUS usage. And therefore, it is uncertain whether the trends in Afirma suspicious rates in this small subset of AUS cases can be extrapolated to an overall AUS usage. Also, the analysis of Afirma suspicious rate was limited to the aspirates classified as AUS on repeat FNA, and which represented only 25% of all AUS samples during the study period. Authors used laboratory average Afirma suspicious rate as a set point, and this particular target is not necessarily applicable to all labs. And further studies to establish broadly expected suspicious rate for AUS nodules are needed. Coming to the conclusion, authors have proposed the use of molecular testing results in conjunction with indeterminate FNA call rates of thyroid nodule as a tool to help explain cytopathologist practice pattern, as well as provide a direction for bringing individual diagnostic category used in line with lab averages or published benchmarks. However, further studies investigating the effectiveness of this feedback, as well as diagnostic patterns seen in other labs or practice settings, will be useful to validate this conceptual framework presented here. Thank you all for your kind attention. And if you have any questions, please feel free to email me. My email address is provided here. Thank you again.